So, a short introduction to and navigating to my pathomation. So, you you can start from Google search, search for my pathomation. Then you get to this URL for my pathomation. You click it, and um, so you get logged in into your account. So the first thing you want to look at is a description of what we want you to review. So come to the quizzes. You'll find after you click the quiz, you'll find two quizzes. One is labeled bounding box quiz. The other one is image classification quiz. So if you click under action, so if you click end it, you'll be able to see basically what we have requested you to do. So you are told to navigate to the slide box option. So if you look to your left, you will see an option labeled the slide boxes. So navigate there and click on the option labeled slide box. Then under slide boxes, select the slide box labeled collection 1. You will find 30 microscope images acquired from gastric tissue from human subjects. Your task will be one, for each image, label it as malignant or benign. And number 2, draw bounding boxes in image sections that appear to you as clear malignant areas. So what that means is go to the quiz on your left, click it, then you find two slide boxes. One is called bounding box quiz, and the other one is called image classification quiz. So under bounding box quiz, sorry. I said you should go to the slide boxes, not quizzes anymore, because now you know. So go to the quizzes. So you will find collection 1 and collection 2. So under collection 1, on your right side, you will see an icon that looks like an eye. And if you bring your cursor to it, you see view slide. So click there, and you will be presented with the slides. In total, there are 30 slides. And so the task is to, of course, now view them. So click the annotate button so that you can be able to expand that image. And then you can zoom in, analyze the image. So the thing is, you are supposed to do two things. After you examine the image, you are supposed to determine whether the image contains some malignant areas, or if not malignant. So you are supposed to label the image as either malignant or Benin. So to label, you can you can click the text icon, and when you click somewhere, you can write. So let's say I think this is a malignant image. It contains some malignant cells or things like those. Then I label it as such, malignant. Okay. And then areas that appear as clear malignant areas, I can draw a box. So I come to this icon, draw a rectangle. Of course, I can choose the right color, maybe green, blue. So perhaps blue would be a good color. Then come to the draw rectangle. And let's say this legion, here looks like contains malignant cells. So I draw a rectangle there. If there is another region that maybe this region, I draw a rectangle there. So of course I can zoom to other places and see. So and um if I think the task is done, now I exit my annotation window by clicking this. But for now, since this was for demonstration, I will delete my annotation. So and then I close this. Then I would go to, so under slide 1, we have section A and section B. So you do the same for section B. So this is section A, section B. After that, you go to slide 2. You get section A, section B, section C, and D, and E. So section A, so if you click annotate, you are presented with that image. Can you see any malignant cells here? If so, you label the image as malignant. And you can draw a box where you see cells which are malignant. So that is the task under quiz 1. Under quiz 2, so you go to the quiz to see what it says. Under quiz 2, this is image classification. So image classification. So if you want what the task that we are requesting you to do is navigate again to the slide boxes option on the left side of the window and click on the option labeled slide boxes. Then under slide boxes, select the slide box labeled correction. They should be correction 2. I think there is a mistake there. Should be correction 2. You will find 30 slides, 30 microscope slides. I think we'll need to edit this so that it is slide correction 2. What I mean is come to the slide boxes And then under collection 2, not collection 1, come to view. So you click view slide boxes. And there you are presented with the slide boxes. So there you find the different images and the slides. So like this one, section A, if you come to annotate, these are small images. We cropped them into small regions for purposes of, we've developed a AI model, but it takes small image, small images of small sizes in terms of pixels. It takes 224 pixel by 224 pixel image. We feed into that model, and it can classify an image as malignant or non-malignant. We trained it using a different data set we obtained online with images that were labeled as malignant and benign. But then these images are the ones we want to test and see whether it can classify them correctly. Are the images we have acquired from the Meru Teaching and the Referral Hospital. We cropped them so that they are small images. So what we want is for you to see whether you can classify them as benign or malignant, because then we will take the same images to the AI model and see the kind of responses we get. So here essentially is for you to come and look at the image. I don't know whether it will you will be in a position to make that a correct classification, but try and see whether if you can just label it as benign or malignant. If you can see reason why you want to classify the image as malignant, just label it. Otherwise, label it as benign. So this is just a classification. So you give it either a malignant or benign label, because then those are the answers we will like use to compare the prediction of the AI model that I'm talking about. So I hope that is clear. So come take a text box and write benign or malignant based on your observation. So do that for the images that are in this window. So we have images, we have slide. So we have again, I think we have as many slides as 30 slides, I believe, because I can see 20, I think we have 29 slides. Yeah, so try to label them as such. So that's basically the task. In case you have, in case you want more clarification, let me know. Thank you.